Hi students, in this video we are going to discuss about Southern blotting, stepwise procedure and application that reveals information about DNA such as size and abundance, and it can be used for the detection of specific DNA sequences in DNA samples. The first step in Southern blotting is the digestion of DNA samples with the proper restriction enzymes. The restriction enzyme, also called restriction endonuclease, it is an enzyme that cleaves DNA into fragments near specific recognition sites within molecules known as restriction sites. Once the restriction enzyme is added, the samples are incubated at 37 degrees Celsius overnight. Restriction enzymes recognize a specific sequence of nucleotides and produce a double-stranded cut in the DNA. Therefore, various DNA fragments of different sizes are obtained. Then, the DNA fragments produced by restriction endonuclease digestions are separated by gel electrophoresis. For gel electrophoresis separation, loading buffer is added to the DNA samples and it is used as a tracking dye which migrates in the same direction as DNA, allowing us to monitor the progress of separation. Agarose gel electrophoresis is most commonly used to separate mixtures of DNA fragments of varying sizes. During electrophoresis, a molecular weight size mark known as a DNA ladder is commonly used to determine the size of the DNA fragments in the samples. The DNA ladder is added to well in the one end of the gel. Once the molecular weight size marker is added, the DNA samples are loaded into another wells. The electric current is applied to pull the samples through the gel. Based on their charge and size, the molecules will travel through the gel at different speeds, allowing them to be separated from one another. The phosphate backbone of the DNA molecule is negatively charged. Therefore, when placed in an electric field, DNA fragments will migrate to the positively charged because all DNA fragments have same amount of charge. Some fragments move through the gel faster than the larger ones due to its small size. After the electrophoresis is complete, the molecules in the gel can be stained to make them visible. When the gel is stained with an intercalating dye such as etherium bromide, the DNA fragments can be seen under the UV light as bands each representing a group of same size of DNA fragments. After the separation of DNA molecules, the double-stranded DNA fragments are denatured with an alkaline solution consisting of sodium hydroxide. The gel is soaked in an alkaline solution with a gentle agitation. At an alkaline pH, hydroxyl ions are predominant. Consequently, guanine and thymine exist at negatively charged. This process breaks the hydrogen bonds between the oligonucleotides, causing the separation of the two DNA strands. After the denaturation treatment, the alkaline solution is removed. Then, the neutralizing solution is used to neutralize the pH of the gel. This allows the more efficient transfer of DNA in the Southern transfers. Now the gel is neutralizing filter paper or membrane prepared for the Southern blotting. To transfer the DNA fragments to the membrane, a transfer buffer, a solid support, and a sheet of blotting paper act as a wick for transfer solution are used. The wick is placed over the solid support, so the end will be in the transfer buffer. Then it is wetted with the transfer solution. Next, pieces of extra blotting papers are placed on the top of the wick. Then they are wetted with the transfer solution. Once the gel is neutralized, it is placed on the wicking paper. Then a sheet of a nylon or nitrocellulose membrane with the same size of the gel is pre-wetted with the transfer solution and placed on the top of the gel. Next, pre-wetted pieces of extra blotting paper are placed on the top of the membrane. They expose the wick or covered with the plastic wrap to prevent transfer buffer from bypassing the gel during the transfer buffer. Finally, a dry stack of paper towels is placed on the top of the membrane and the gel, and a glass plate is placed on the top of the sheets of paper towels with a weight to maintain a tight contact between the gel and its membrane. Buffer transfer by capillary action from a region of high water potential to a region of low water potential is then used to move the DNA from the gel onto a membrane. Consequently, ion exchange interaction by DNA into the membrane due to the negative charge of the DNA and positive charge of the membrane. The transfer is allowed to proceed overnight, and once it is completed, the blotting material and the membrane are carefully removed from the gel. Next, the membrane is briefly rinsed to remove any particles that may be stuck during the transfer. Then it is exposed to UV radiation to permanently attach the DNA to the membrane. After attachment of the DNA fragments, the membrane hybridization and radio-labeled DNA probe is performed. The membrane is placed in a bottle containing a pre-hybridization buffer solution, which is used to reduce non-specific hybridization with the probe. Next, the bottle is incubated in hybridization oven at 42 degrees Celsius for 2 hours. Once the incubation is completed, the pre-hybridization solution is removed. Then, hybridization buffer is added into the bottle. Next, labeled DNA probes are added into the hybridization solution. The probes are fragments of DNA with variable length which can be radioactive fluorescent labeled. Once the hybridization probes are added, the bottle is incubated overnight in hybridization oven at 42 degrees Celsius. DNA contains a large quantity of phosphorus in phosphodiester linkages between bases and oligonucleotide chain. DNA can therefore be trapped by placing its non-radioactive phosphorus with radioactive phosphorus 32. The radioactively labeled DNA probe hybridized to their complementary sequences in the DNA fragments. After hybridization of each probe to its target sequence, the hybridization solution is removed. Next, a wash buffer is added into the bottle. Then, the membrane is incubated at 52 degrees Celsius for 30 minutes. The washing process is repeated 3 times to remove unbound and weakly bound probe. After hybridization, autoradiography method is carried out to identify the location of radioactively labeled DNA in the membrane. The southern blot filter is placed inside lightproof box. Then a X-ray film is laid over the top. The lid of the box is closed and left for several hours to several days. The radio-labeled DNA exposes the film, which will show a black band that indicates the positions of labeled DNA in the membrane. Subsequently, these informations can be used to determine which gene is present in each sample. Also, the position of each gene can be identified in the gel. Consequently, after electrophoresis, the bands of interest can be cut out from the gel so that each gene can be isolated for further analysis. Thank you.